Hey team, Dr. Jake Order here. Now I'm going to take you through a technique in this video, cell culture. Now there's a lot to cover in cell culture, but I'm going to try to keep it pretty short, pretty snappy, just to give you some of the basics of cell culture. Here we have some cell cultures here, cells growing on a dish here, and we can see them nicely because of phase contrast microscopy. One of the cool things about cell culture is you can actually view your cells under a microscope growing live. But I'm getting a little bit ahead of myself, so let me jump back into it. So cell culture is when we grow cells in a flask, basically. So we can grow them in big flasks to get a lot of cells up to a big number. We can grow them in smaller flasks if we want to do things like view them under a microscope, or we can grow them in these very tiny wells in a plate. And this we can do high throughput drug screening, for example, where we can test loads of drugs on a process that's going on in the cell. So we can initiate a process in the cell, for example, like inflammation, and see if we can inhibit it with hundreds of drugs. I've done quite a bit of this in my time trying to develop new drugs to treat disease. So you can hypothesize that a particular cell process contributes to a disease. You can mimic that cell process in cells in a dish, try and inhibit it with drugs. It's a fantastic way to essentially reduce some of the other research that might involve animals because you're doing most of your testing in cells in a dish. It's fantastic. Now, if I can just pop back, there's a couple of different ways to grow cells. So in this case, these cells are adherent, which means that they stick to the bottom of the plate. Now, I'm going to use a classic example from a video that I've covered previously, is if it was a monocyte is a circulating immune cell, so it doesn't adhere. If we were to grow monocytes in growth media, they would just float around and they wouldn't adhere. Now, monocytes, when they leave the blood vessel, can turn into macrophages, which sit in the tissue. Macrophages are adherent cells because they are tissue resident cells. So when we put them in the flask, they will stick to the bottom. So these could be macrophages here because they are sticking to the bottom. And so that's one of the two major ways to break up cells, adherent and non-adherent cells. There's other ways to break up cell types, and this is probably the two big divisions of breaking up cell types. One is primary cell culture, the other one is immortal cell culture. So primary cell culture is when we take an organism and we take some cells from them in some way, and then we can grow those cells quite temporarily in addition to experiments on them. These are actually my white blood cells. These are my neutrophils. There's some lymphocytes there. Here's a basophil. Sorry, an eosinophil. Oh, naughty. That's an eosinophil, sorry. Neutrophils, lymphocytes. These are my white blood cells there. So those are primary cells. I took my blood, I took the white blood cells out of them, viewed them under a microscope. That is primary cell culture. Then there is immortal cell culture. And it's when we have edited those cells, or perhaps nature has edited those cells in a way that they will just keep dividing and dividing and dividing. And so what I can do is I can give you a sample, you can ship your sample over to your friends in the UK, they can ship a sample to America, and we can all grow the same cell type, and they will divide and divide and divide. So that's an immortal cell culture. So let me run down some benefits, cost-benefit analysis of these. Primary cell culture are accurate, right? So if I take my neutrophils out of my blood, they are very accurate representations of my neutrophils because they are neutrophils, right? So it's an incredibly accurate model to replicate the cell type that you're interested in. However, they're difficult to obtain. You need human ethics to take cells from humans. You need animal ethics to take them from animals. They're not as easy to obtain as immortal cells, which we can just send around the world, like I just mentioned. They also have a limited lifespan. This is called senescence, and essentially cells can only divide a few times. Normally, it depends on the cell type. Neutrophils don't divide at all. They die within 24 hours, so they're very hard to do primary cell culture on. But other cells can divide maybe up to 30 times before they would just stop dividing. And this is a bit of a difficult one. They're less repeatable, right? Say I was doing human primary cell culture. I would sample from the humans available around me. Often it's lab mates that you go on a rotation system after signing all the ethics and everything like that. And so they are often people very similar to me with my genetic makeup, the Tasmanian perhaps, not my genetic makeup hopefully, but similar to me in many ways. And so it's quite difficult to replicate. They're the same age as me often. And so if another lab was to try, they might have a different spectrum of genders, of ethnicities, of nutrition. And so they might get different results to the results we got because of where we got those samples from, right? So there is some issues of repeatability with primary cell culture, but in many respects that might be a good thing, because if you want to take that into clinical trial, you need to test it amongst a diversity of genotypes and phenotypes and ethnicities and diets and all that kind of stuff. So that might be a good thing as well. Immortal cell culture. Well, just to explain, how do the cells get immortalized? So there is a few ways. One, we could genetically engineer the cells so they don't undergo senescence, so they keep dividing and dividing and dividing. Now, you might recognize that dividing and dividing and dividing. It sounds a lot like cancer. So that's another location that we get this from. We get it from cancer tissue. So if someone has a cancer of the cells that produce monocytes, and so they divide and divide and divide, a form of leukemia, then if we take those cells, they will keep dividing and dividing and dividing, and they will look like monocytes, right? Now, there's actually really horrible stories where tissue samples were taken from people. One of the most famous is Henrietta Lacks, who was an African-American woman, and her cancer cells were taken without her permission, and they have now created a cancer line that's called the HeLa cell cancer line. Now, the HeLa stands for Henrietta Lacks, and they are everywhere. We've got some up in the lab. They are ubiquitous throughout the world. They have led to amazing discoveries, which is a tough ethical balance, but the way that they were taken is definitely unethical. And there's fantastic books out there on this story, and I highly recommend you go read it because it's got a lot of issues there: racism, power imbalance, feminism issues there. It's a really great topic to dive into, and I highly recommend learning more about it. So, two main sources. One, we take cells, primary cells that we edit, and then they become immortalized with genetic engineering, or we can take what nature has edited randomly with mutations in the form of cancer. And so we can take those cancer cells and we can keep growing them. So there's a few things there. One of the drawbacks is, what is it really? Right? So one of the first things to go in cancer cell lines is the DNA repair mechanisms. This allows the cancer to build up mutations, which is required. You need a number of mutations to become a successful cancer, right? So those mutations continue to accumulate throughout cell culture, and you end up with something that you're not quite sure what kind of cell type it is. The other thing, for a cancer to become metastatic, it has to lose its identity a bit. If a lung cell ends up in your liver says, that cell's not supposed to be there, and they signal for it to die. But if that lung cancer cell mutates so it doesn't even look like a lung cell anymore, it might be able to infiltrate into the liver, and your cancer might be able to become metastatic. It might be able to spread. So part of becoming cancerous is losing your identity. So is it still a monocyte if it's a cancer monocyte line? And we certainly know that there are certain things you cannot replicate in cancer cell lines, cancer immortal monocyte cell lines that you can replicate from primary cell culture. So it certainly loses some identity. A huge example of that is those HeLa cells I was talking about. A human cell should have 46 chromosomes. HeLa cells have crazy numbers of chromosomes, like 80 or 90 chromosomes, depending on which strain you got, which lab you've got. I should say this sample was taken decades ago. Several decades ago. And so it's sort of an immortal cell line that's gone well beyond the death of Henrietta Lacks, in fact. And so it is mutated massively. And so, what really is that cell anymore? And people don't really treat the HeLa cell as a cell type. What they normally do is insert genes of interest and genetically engineer the cells to express things that you want to investigate. So even though it may not have a pattern recognition receptor because it is not a macrophage, you could genetically induce the expression of a pattern recognition receptor in that cell. And so now it's got that pattern recognition receptor for you to investigate. So that's a lot of the kind of things that we do with immortal cell lines. Very cheap. So once you buy it once, it can be a bit expensive, maybe 800 Australian dollars to buy a cell line. You can grow it up as much as you like, share it with friends, and so it's very cheap to grow immortal cell lines. Whereas primary cell lines, you've got that human ethic issues, but also if you take them from animals, animals are very expensive, and so yeah, immortal cell lines are much cheaper. They pretty much have an unlimited lifespan. There's a lot of asterisks on that, right? And we have what's called a passage or a passage of cells, which is when we, if a cell's dividing and dividing and dividing, we don't want that many cells, so we ditch half of them and then give it new nutrients to grow, and then it'll divide and divide and divide, and then we ditch half of it. Normally after about 30 passages, the cells will start to act very weird. So what we do is when you first buy the cell, you grow up a huge number of cells, then you freeze them down. And if you do that, you can then sample back from the original divisions a fresh batch of cells whenever your cells start to act a little bit funny after a lot of passages. And it's quite repeatable-ish, right? So I mentioned the problems with HeLa cells, but I can replicate other people's experiments using the exact same cells that they had. Now, there might be some genetic drift going on there, but normally we don't have a problem. If I buy it from a reputable seller, the same seller that they bought it from, ATCC, for example, as a company that sells cell lines, if we buy those cells and they buy those cells, I can replicate work that's been done in the UK or Japan or Singapore or America, for example. Whereas if it was humans, there's a bit of that variability I talked about. So what is that media it's growing in? Well, it's a mixture of salts. It's a mixture of, potassium, calcium, sodium, chloride, all the salts that you will find in the human body. There's also amino acids, there's sugars. Interestingly enough, it's quite hard to replicate all the nutrients that you need to grow cells. So we typically supplement this media with serum, which is the fluid component of blood from fetal cows. Now, fetal cows are typically sterile, which is one of the reasons why we take the blood from the fetal cow. We get the serum, and then we can mix that serum with our growth media, and then we can grow our cells. So one of the things going on there is that growth media contains all the nutrients you might expect, but then there's a lot of extra stuff, and it's hard to get everything in there. So we take the serum from a cow, essentially, to put into the media. So now it's full of the fluid component of blood, which contains all those other things, proteins, growth factors, all sorts of stuff. Why is it red? Well, we put phenyl red in there. Now, phenyl red is a pH meter, so it changes color on pH. So here it's gone acidic. And we do this for a very simple reason. pH tells us if the cells are starving or infected. Basically, metabolism produces H+ ions and makes it acidic. ATP hydrolysis produces H+ ions. So metabolism makes it acidic. Now, if we don't change the growth media for a very long period of time, the acid will build up, and we can see that we haven't fed our cells in a long time with fresh growth media, and so it's gone yellow, so we need to change our growth media. If our growth media changes really quickly, we know that there's a lot of metabolism has happened, much more than it should have in a short space of time. Now, if a lot of metabolism happens quickly, that's a surefire sign you've got an infection in your growth media. So obviously what you grow your cells in is very delicious to bacteria and fungi. So if you don't use proper sterile technique, you'll get an infection and you'll end up with loads of bacteria, and you'll end up with your wells turning yellow very, very quickly. I can honestly say that hasn't happened to me. I haven't infected cells. And it's one of those things that I'm going to lose that record one day, but I'm holding on to it very strongly right now. I'm very proud of myself. All right, so what are the problems with cell culture? Why don't we just cell culture everything? Why do we ever use animal models or even human models? Surely we can just use cell culture. Problem number one, cells don't live in isolation, right? So normally we're growing these cells, just macrophages or, just muscle cells. Cells don't grow like that. Now, as an example, I'm going to talk about a disease that was in my previous few videos, Alzheimer's disease. So Alzheimer's disease, the leading hypothesis of Alzheimer's disease is the amyloid hypothesis, which states that amyloid builds up in your brain, it sticks together, forms oligomers and clumps, and those lead to pathological processes. The amyloid builds up in your brain, possibly because you've got too much amyloid precursor protein in your neurons, that's one cell type, or you've got too much enzyme that chops up the amyloid precursor protein the wrong way, so now you've got overproduction. Perhaps you have under removal. Now, amyloid is removed through the blood vessels. Now, blood vessels have endothelial cells, pericytes, smooth muscle cells, and your blood. I mean, you can't even count that as one. Oh boy, lots. Okay, but another way is amyloid is also phagocytosed by microglia. That's another cell type. There's another cell type that could be critically involved, astrocytes. They're the support cells for neurons. There's lots of evidence, stuff going on with astrocytes. We also know that peripheral immune cells are coming into the brain in response to Alzheimer's disease. Some have been implicated, such as T cells and neutrophils, but there are even more. What does the amyloid do once it's in the blood? Well, it gets processed by the liver, which is hepatocytes. And then we have lipoproteins that are going around the blood. What are they being produced by? But anyway, you can see that at absolute minimum, we've got 11 cell types working with complicated structures in organized fashion. So not all just mixed together, right? The pericytes, endothelial cells, smooth muscle cells are all on this blood vessel, right? All of these cell types and all of these structures could be contributing to Alzheimer's disease. We also know that obesity increases your risk, so that's fat cells. We know high blood pressure, so now you've got to model some sort of blood pressure, high cholesterol, so now you've got to model the cholesterol. All these processes contribute to your risk of Alzheimer's disease. How are you going to model all of that in a dish? The answer is we can't. We absolutely can't. And so to model the complete disease, we cannot use cell culture. We could take a small part of it. That's what I did. I took just the microglial response over here to amyloid and I modeled that in a dish. You can take a tiny part of it and model in a dish. You cannot model the whole disease. And so we could do some of our drug screening in a dish to figure out perhaps we need a drug to inhibit this enzyme, perhaps we need a drug to prevent inflammation, perhaps we need a drug to transport the amyloid out to the blood better. You can model those kinds of things in cell culture. You cannot model the whole kit and caboodle. And so to understand what the drug does to the whole disease in general, we need animal models as an early indicator, and then we need human clinical trials after that. So yeah, all of that's to say our cell culturing techniques aren't sophisticated enough to replicate the entire body in a dish, right? So we kind of need animal models, unfortunately. It's a real pain, and I really can't wait until we get beyond it, but we're not there yet, that's for sure. Another problem. We're growing these cells in flat dishes. So the cells often adhere to a flat dish, but the reality is cells live in a three-dimensional space supported by extracellular matrix, right? So that's another problem. The vast majority of cell culture is in flat dishes. The solution is jelly. Now, this is a growing field. It's very cool, and it's made some amazing discoveries. And essentially, it's jelly, right? Like gelatin with cancer cells in it. So you can create these extracellular networks. Here's the scanning electron microscopy of it. So you can see it's a mesh of proteins, glycosylated proteins and polysaccharides, exactly like your extracellular matrix, also exactly like literally jelly, because jelly is made essentially from extracellular matrix. Yeah, so essentially we can grow cells in jelly in extracellular matrix to replicate that 3D environment. Now, when we do that, we immediately notice it changes the physiology. So by sticking to a glass or a piece of plastic, we're changing the physiology and we're getting away from replicating what was going on in the animal and in the human body. And so by suspending them in a mesh of extracellular matrix, we get more realistic responses and we get different responses, which is very, very interesting. Now, this process led to one of the coolest discoveries. Honestly, this is really sweet. It's got thousands of citations, this paper. Matrix control of stem cell fate. This is an amazing discovery. Very modest title here. But this is what they did. They took stem cells. Stem cells can turn into other cells, obviously, and they put them in a 3D cell culture matrix, which is basically jelly matrix. So we call this 3D cell culture because it's making up a 3D cell space, whereas flat cell culture we call 2D cell culture. So this is three-dimensional cell culture, and it's in a jelly matrix. Now what they discovered was you put the stem cells in the jelly, and you make the matrix really stiff by having a high concentration of the proteins that aggregate together tightly to make a stiff matrix. And the stem cell turns into a bone cell, right? So somehow the cell was detecting the rigidity of the extracellular matrix, and they said, I'm in a very rigid matrix. That probably means I'm in the bone. So as a stem cell, I should turn into a bone. They then made a mediumly stiff matrix, a bit more softer, and the cells turned into muscle cells. So the cells were like, okay, the rigidity of this extracellular matrix feels like a muscle cell, so I should turn into a muscle cell. And lastly, they found if they put it in the most soft, you can even suck this up into a pipette. That's how soft it is. It is such a soft, squishy matrix. Then the stem cell turns into a neuron. It turns into a brain cell. It says, I'm in such a squishy, gelatinous mass. It must be the brain. So then they turn into a neuron cell. So it turned out the rigidity of the matrix helps contribute to the fate of stem cells, what they turn into. Now, this isn't a complete picture. There's also growth factors that can turn you into different kinds. Like, how do you know you're a heart cell versus a skeletal muscle cell and this kind of thing. So there's a lot more going on, but certainly a major signaling pathway is just the rigidity of the 3D matrix, which is, again, it shows you that limitation by growing cells on a dish, on a 2D plane. Cells grow on 3D planes, and there's very important physiology going on there that we are missing by planing our cells out on a 2D plane. Brilliant, guys. That was just a real brief introduction. I could talk for hours about cell culture. But yeah, that was a brief introduction to cell culture. If you have any questions, pop them in the comments below. I'll be happy to reply. Like and subscribe. Follow me on Twitter. All that jazz. Catch you later.